All right, a little bit of review first, and we're going to hit that Gibbs free energy stuff hard today. But first of all, just a little review. Energy, if we define it, we said it's the ability to do work or to make things move. We talked about, at least in terms of biology, most all the energy on Earth that's important for life comes from the sun. We're going to say it's the ultimate source of Earth's energy. We talked about different kinds of energy already. When we think about kinetic, we ought to think about what? Movement. In physics today and yesterday, we're talking about the same kind of stuff in physics. But we're talking about the mathematical side of it. There's an equation to calculate how much kinetic energy something has. And it turns out it's 1/2 mv squared. What do you think the m stands for? Mass. And the v? Velocity, speed. So that's kinetic energy. Potential energy, on the other hand, is energy that's what? Stored. Now, in physics class, we're talking about gravitational potential energy. Energy stored in something due to what? Its position above the ground. In this class, it's not so important. In this class, we're talking more about chemical energy, stored mostly where? In the bonds of the ATP is good at storing energy. But we're going to talk in a minute about, you only have about 20 seconds worth of stored ATP. Why? That sounds kind of dangerous, right? I stressed to you in this class that your body is conservative by nature. It stores stuff. It prepares for rainy days. But you've only got 20 seconds worth of ATP stored up. And if you run out of ATP, what happens? You die, and there's no coming back. So it seems like 20 seconds worth is not very smart. That kind of sounds like the federal government, right? Why? Why only 20 seconds worth? Turns out that ATP is really reactive stuff. And the hydrolysis of ATP is what kind of reaction that you studied about in your Moodle assignment? It's spontaneous. It happens on its own. And it turns out it's not only spontaneous, it's fast and spontaneous, which means we're going to store it. So if we want to store energy for the long term, we have to store it as what? As fats. Maybe glycogen also. Bring me the key back. We also talked the other day about the laws of thermodynamics. First law of thermodynamics, we also call the law of conservation of energy. And it's an idea that you've all heard for a long time. You can't what? You can't create energy, you can't destroy it, but you can convert it from one kind to another. A lot of you on the last test, when I asked you for the function of the mitochondria, you said it creates energy. And you got a big X on your paper. It doesn't create energy. It takes energy from where? Mitochondria. It takes energy from food and puts that energy into ATP. A lot of you want to ask about the function of chloroplasts. You said it makes energy. And again, you got a big X. It takes energy from the sun and puts it in sugars. You can't make energy. You can't destroy it. You can convert it from one kind to another. But a big part of our discussion today is going to be about the second law of thermodynamics. It's kind of confusing at first. You're going to have some math to deal with the second law of thermodynamics. But don't let this get you bogged down too much. I'm going to talk about the second law of thermodynamics in 3 different ways. They're all really saying the same thing. Sort of like with osmosis. We talked about it like 1,000 different ways, right? They all meant the same thing. First of all, I'll go basic. The second law of thermodynamics basically says that what is constantly increasing in the universe? Entropy, which is a fancy word for what? Chaos. Disorder, chaos. And at first, you might think that can't be right. Because all we've talked about in this class is about, in terms of evolution, is that life started out as what, according to the theory of evolution? Before that? goo, right? Soup, organic soup, amino acids and such in water. And somehow that soup formed what? Bacteria, prokaryotes. And the prokaryotes, through endosymbiosis, formed what? Eukaryotes. And then evolution eventually led to plants and animals and fungi and all these other things. That doesn't seem like entropy. That seems like what? That seems like getting more and more order, not more and more chaotic, right? But we're only looking at a small piece of the universe. To do all those things, what had to be used? Lots of energy. And every time you take a bite of a candy bar and you break down that sugar and you do cell respiration on that sugar, some of the energy in that sugar is used by your body to do stuff like talk and move and walk and think. But a lot of that energy ends up where? Giving off as heat, waste. And once it's given off as heat, what can we say about it? We can't get it back. It's gone. It's not useful anymore. Where does it ultimately end up? Somewhere in space, never to be used again. Another way of thinking about entropy is that the amount of usable energy is constantly what? Decreasing. The amount of usable energy on Earth is constantly decreasing and becoming what? Heat. Waste, heat.
So does that mean at one point in time in there, we're still running, not going to be any more energy?
Eventually it's going to run out, right? Another way to think of it is that the amount of energy in the universe is finite. It's limited. It's not infinite. It's finite. There's only a certain amount. And once it's gone, it's gone. Now, we don't need to worry about that because there's way more than we're ever going to use, right? But eventually, probably millions of years down the road, it runs out at some point.
How do they keep a measure of how much energy?
I mean, there is no way to do it. I mean, they're guesses. Like they guessed how long hydrogen in the sun will last, when the sun will go out. But it's a guess. I mean, we don't really know. Well, the sun is energy, right? So, hard to say. Another way to think about the second law of thermodynamics is that in order to overcome entropy, in order to stay ordered, we must constantly use what? Energy. In order to overcome entropy, there must be a constant source of energy. And that's kind of what that bottom little blurb there says, right? Energy must be added to maintain order or homeostasis. To keep from falling apart, to keep ourselves organized and ordered, you get it? We must use a lot of energy to do that. Now, we'll get into the math here in a second, but just a reminder about a couple of important words and ideas. Everything you do is metabolism. Let's say you look over at, who am I going to pick on today? Let's say you look over at Andrew, and you just think, hey, I really don't like Andrew. How many of you have that feeling? Okay, you just look over at Andrew and say, hey, I really don't like Andrew. What makes you have that feeling? Chemical reaction. Maybe he's giving off some funky pheromone, right? We don't know, okay? But regardless, it's chemistry. Everything you do is chemistry. Thoughts, memories, movements, they're all chemistry. What now? Oh, yeah. Regardless, all those chemical reactions put together are called our metabolism. That's a good definition up there at the top for metabolism. The sum of all the chemical reactions that are in organisms. Everything you do makes up your metabolism. All your chemical reactions. Feelings, thoughts, memories, movements, digestion material, cell respiration. It's all chemistry. Now we're going to take metabolism and break it down into two main versions of metabolism. Catabolism or catabolic and anabolism or anabolic. We talked about these two words before, several times. You should already know that catabolic means to break things down. Hydrolysis reactions. And typically, when you break a bond, what happens? Energy is released. So catabolic reactions tend to be exergonic reactions. And that's an important word for today, exergonic. Reactions that what? Release energy, give it off. So again, catabolic reactions break things down. They're hydrolysis reactions. They tend to release energy. Maybe heat and maybe other forms of energy. They tend to give off energy. If they're releasing heat, they're increasing what? Entropy. They also tend to increase entropy in the universe. On the other hand, anabolic reactions. Those are synthesis reactions. What are two other words we use to describe synthesis reactions? Condensation and dehydration. Both meaning the same thing. You'll be able to recognize all those terms. Synthesis, dehydration, condensation, anabolic. They all mean building up. If you're building up things, if you're assembling molecules, what does it take to make a bind? It takes an addition of energy. You've got to add energy. So sometimes we call these reactions endergonic. They take in energy. They need energy to make them happen. And since they're taking in energy, what are they doing to entropy? Since they're putting things together, they're decreasing entropy. They're making order. But you've got to add energy to do that. It takes energy to get order. Does that make sense? Everybody good there? Another really important idea is this idea of coupling. If we use the word couple like a verb, what does it mean? Put together, right? To join. Just listen to what I'm saying and don't write anything here for a second. Your body, all your body's endergonic reactions, they have to be powered by something, right? They have to get their energy from somewhere. Where do your body's endergonic reactions typically get the energy they need to happen? From ATP. From what about ATP?
Phosphate, for example.
Yeah, and the word for that is?
Phosphorylation.
Let me know. The hydrolysis of ATP, the breakdown of ATP. You're thinking phosphorylation. The hydrolysis of ATP. So here's how I'm going to define coupling. Coupling is when an endergonic reaction has to be paired up with what? An exergonic reaction. And typically the exergonic reaction it's paired up with is which one? ATP hydrolysis. So coupling is when an endergonic reaction is paired up with an exergonic reaction. That's almost always ATP hydrolysis. Because that exergonic reaction provides what? The energy needed to make the endergonic reaction happen. Does that make sense?
What was the definition of energy?
It's when an endergonic reaction is paired up with an exergonic reaction. Usually ATP hydrolysis. And that provides the energy to make that endergonic reaction go, to make it work. Everybody good with that? All right, a little bit more about ATP here. I'm hoping everybody knows, first of all, ATP falls into which of those four groups of macromolecules? Nucleic acids. Which means it's built out of what? Monomers. Nucleotides. Now, I want to look at a picture of a nucleotide first. Here's ATP. What is always the center part of a nucleotide? Sugar. And in the case of ATP, it's ribose. The same sugar we would find in RNA. Over here, what is that part of a nucleotide? It's the nitrogenous base. And in ATP, there's only one nitrogenous base. What is it? Adenine. A. A is the only one. And then the other part of a nucleotide is a phosphate group. In this case, there's 3 of them. Now this picture, if you understand this picture, you know a lot. This picture has a lot of good information in it. First of all, going this way around our circle, this reaction is supposed to be what? What's happening between here and here? Yeah, and the word for that is hydrolysis. ATP is being hydrolyzed. This bind right here is being broken. And typically that phosphate is added to something else. That phosphate is broken off of ATP and transferred like to an enzyme or to another protein. And what does it do once it's transferred? It gives it energy and usually does what to it? If I gave Christina some energy right now, what have I done to Christina? I've activated her to do something, right? I've turned a switch on and made Christina's going to do something now. We call that phosphorylation. When ATP is broken and that phosphate is transferred to something else and it gives it energy, it gives it activation energy, it makes it work. But notice what it says right here, important statement. ATP gets broken down and that provides the energy for your body's energonic reactions. It provides energy for your body to make proteins. It provides energy for your body to conduct nerve impulses. It provides energy for your muscles to contract. So the breakdown of ATP provides the activation energy to make most of your body's reactions work for you. Once we break it down, though, we're left with ADP and P. Think of those, and I've given you this analogy before, like what? Those two pieces we could compare to what human device? A battery, but a battery that's what? Dead, but capable of what? Being recharged. And in your body, what's the process of recharging your batteries? Cellular respiration. Cell respiration is going to take this dead battery, recharge it, using energy from where? Food. That's the reason we eat. Get energy to recharge our ADP and P batteries so that we're back to ATP and now we can carry on our body's energonic reactions. Does that make sense? If you understand that slide, you know a lot of stuff. It's when a phosphate is removed from ATP, transferred to some other protein or enzyme, and when it's transferred, it takes energy with it, so it provides energy to that protein or enzyme. So phosphate's removed from ATP, transferred to another protein or enzyme, and it provides energy to that protein or enzyme. All right, couple other quick things. I think I've covered pretty much everything on this slide. ATP hydrolysis is said to be spontaneous, which means what? It happens on its own without any addition of what? Energy. Does spontaneous necessarily mean it happens fast? It just happens. Like rust, for example, like a piece of metal rusting. That's spontaneous. But does that happen very quickly? No. But I want to tell you that ATP hydrolysis is spontaneous and what? And fast. That's why we can't do what with it? That's why we can't store it. That's why we have to continually do what? Yeah, we have to recycle, but we also have to continually be doing what process? Cellular respiration. And is ATP hydrolysis exergonic or endergonic? ATP hydrolysis, exergonic or endergonic? Exergonic. It releases energy for other things to happen, right? All right, now let's get into the fun stuff. Let's talk about Gibbs free energy. How many of you folks are AP chemistry folks? Now, you probably went into some gory details of Gibbs free energy, right? We're not going there. I will tell everybody in here, this can get really complicated. We're not going there. We're going to keep it simple. If you understand what I'm about to tell you, you will know all you need to know about Gibbs free energy. The math is easy. The math we're going to do at least is easy. And the idea is not that complicated of an idea. When you read this in your textbook, you might have thought, man, what are they talking about? I'm going to simplify it. I'm going to tell you what you need to know, and that's all you need to know for purposes of this class. If you want to know more, take AP Chemistry. Okay, and it can get kind of complicated. We want to talk first about this concept of free energy. Free energy is energy available to help us to do work. It's energy we can use to make us do work. Now notice there's a couple of other statements up here in this definition. The portion of a system's energy that can perform work when temperature and pressure are held constant. In your body, are temperature and pressure held constant? We call that what? Homeostasis, right? Temperature and pressure are constants in your body for the most part. They may vary slightly, but not very much. So free energy is the energy that can help you do work. It's the energy you can use. It's the energy that makes you do stuff. Now, a guy named Gibbs figured out some of the math here. So sometimes we'll call it Gibbs free energy. Sometimes we might in biology also call it what else? What is the energy that we use to make something happen? It's another word for that. The energy needed to start a chemical reaction, for example. The energy needed to start a reaction. What's the word for that? Activation energy. We're going to use those two words in this class pretty much interchangeably. Activation energy and free energy. The energy needed to make something happen. Now, in terms of this equation up here, the symbol that we use for free energy is delta G, named after Gibbs. Now, delta, we usually write it out how? Little triangle, and it means what? Change in. So we're going to say the change in free energy is equal to delta H, which is the change in what? The total amount of energy in the body. So we're talking biology. The total amount of energy in the body. Another word we'll use for that is enthalpy. Please do not confuse enthalpy with what? Entropy. Enthalpy is the total energy available. Entropy is what? Disorder, chaos. Two completely different things. So you take the total amount of energy and you subtract from that the temperature. It's got to be what kind of temperature? It's got to be in Kelvin. Sometimes it's called the absolute temperature because it's based on Kelvin is sometimes called absolute temperature because it's based on, help me out here, it's Kelvin based on. What's the Kelvin temperature scale based on? Absolute zero, right? Real zero, which means when there's no what? Tell me more. Absolute zero means when there is no what? No heat. Therefore, no what? Movement. Okay? Zero. But we take the temperature and we multiply it by the change in entropy. That equation kind of looks intimidating. The math we're going to do is pretty easy. You got this little handout the other day. If you got it, get it out. If you don't, hold up your hand. I've got a few extra copies. I can give you one. There's a very similar problem in your Moodle assignment. That's why I said to have your Moodle assignment out, because you can check the math here. We're going to work the problem off the handout, but it's almost the same. It's exactly the same kind of problem as the one in your Moodle assignment. A couple of things to know about this equation first, though, before we try to do any math. If we calculate this delta G, free energy, and we get a number for a particular reaction that's negative, what does a negative in front of an energy number mean? But before that, if you get a negative number in an equation, what does it mean? It means it's being given off. It's released. So the energy is released, which tells us it's what kind of reaction. And it's exergonic, right? So there's four things I want you to know about a negative delta G. Negative delta G tells you first of all energy is being released. Number two, it tells you the reaction is an exergonic reaction. Negative delta G, energy is released. It's exergonic. What else did we say? It's spontaneous, which means it will happen without help. It'll happen on its own. And what's the fourth thing? What else does a negative delta G tell you? What do we say about exergonic reactions, Claudio, and entropy? They increase the amount of entropy in the universe. So negative delta G should tell you four things. Energy is being given off, it's being released. The reaction with a negative delta G is exergonic. It's spontaneous, and it increases the amount of entropy in the universe. Now, if we do the same calculation and we get a positive number, it's kind of just the opposite, right? Positive delta G tells us what about energy? It's being taken in. It's required. Energy is a reactant. To be added to make this happen. It tells us it's what kind of reaction? Endergonic. It tells us what about spontaneous or non-spontaneous. It's not a spontaneous reaction. You have to add that amount of energy to make it happen. And what does it do with entropy? It decreases entropy because it increases order. It's putting things together, right? It's organizing things. It's making bonds instead of breaking bonds. So a positive delta G. Energy is needed. It's absorbed. The reaction is endergonic. It's not spontaneous, and it decreases the amount of entropy in the universe. Everybody okay with that? Conceptually, if you know that, that's as deep as we're going. You understand that stuff, that's conceptually as deep as we're going with gives free energy. Mathematically, we're not going all that deep either. Let's take this problem and let's work it. Let's switch over to the camera view for a minute. So this tells us an experiment determined that when a protein unfolds to its denatured state from the original folded state, the change in enthalpy is 56,000 joules per mole. Well, that's delta H, change in total energy, 56,000 joules per mole. Problem also tells us, also the change in entropy, delta S, is 178 joules per mole. And this happens at a temperature of 20 degrees Celsius. Well, first of all, in our equation, we need the temperature, but it's got to be what kind of temperature? Kelvin. So 20 Celsius is 293 Kelvin. And then we multiply that by the change in entropy, delta S. That's 178 joules per mole. But I'm going to tell you, whoever wrote this problem made a slight mistake here. What other unit ought to be associated with this entropy thing? It ought to be joules per moles times what? Times Kelvin, because otherwise the units don't work. There should be a Kelvin down here on the bottom of that fraction. And whoever wrote the problem just forgot to put it in there. Because here's what's going to happen. The Kelvins are going to cancel out, right? And now if we do the math, if we multiply it, let's say 293 times 178. That's 52,154 joules per mole. That's got a minus sign in front of it. 56,000 still up here. So now if we subtract that off, we end up with 3,846. Now we need to get the numerical part. But that numerical part, for our purposes, is not really the important part. What's the important part of that? The sign. It's a positive number, which tells us this process of unfolding this protein is what kind of reaction? Endergonic. To make this protein unfold, what do we got to do? Add energy. We got to add that much energy, right? And because it's an endergonic reaction, what are we doing? It's not what? Not spontaneous. It's not going to happen on its own. It's not going to happen unless what? Unless we add that much energy. It's a non-spontaneous reaction. Does that make sense? I think the Moodle assignment question you had, you ended up with what kind of delta G? You ended up with a negative. If you did it right, you got a negative number, which meant it was what kind of reaction? Spontaneous, and therefore exergonic, and doing what with entropy? Increasing entropy. Make sense? That's as hard as this gets. Everybody in here can do it. Well, I'll say it's as hard as it gets. For this class, it's as hard as it gets. If you've done it, the chemistry stuff, you know it can get much more.
Okay.
Yes.
That problem, if something's denatured, that means the bonds are breaking. Wouldn't that be releasing energy?
Some. But in this case, it takes more energy to break those bonds than they're releasing. It's one of those things where you have to put more in than you get out, kind of thing. All right, a couple other points from this slide that are pretty important. Well, my PowerPoint doesn't like me very much. We'll see if it comes out of that loop. Two important points here. ATP hydrolysis. Please. ATP hydrolysis is a exergonic reaction. It gives off energy. It gives off about 13 joules of energy per mole of ATP. So this is saying that delta G for ATP hydrolysis is about negative 13. If ATP hydrolysis is exergonic, then the making of ATP has to be what? Endergonic. If when you break down ATP, you get a delta G of negative 13. When you make ATP, delta G is probably going to be what? Positive 13. You get out what you put in, sort of thing in this case. All right. Let's see if this is going to cooperate. Apparently not. All right, going to take me just a second to get this fixed up. Eventually, we'll get there. All right, now, let's get back to what we were doing. This is a good example of energy coupling. This reaction down at the bottom. It's what kind of reaction? We said coupling is when you put what two kinds of reactions together. Endergonic and exergonic. Well, we know this one right here is what kind of reaction? That's ATP hydrolysis, right? That's exergonic. Which means its partner has to be endergonic. This reaction, ATP hydrolysis, is providing what to make this one go? This is providing the activation energy, the free energy, to make the bottom one happen. They're coupled. This one down here is what kind of reaction? Other than endergonic, it's also not spontaneous. What makes it happen? The energy from here makes it happen. They're coupled. This provides the energy needed to make it start, which we're going to call what two things? Activation energy or free energy. As difficult as this gets. This is another example. This is showing you that moving, contracting your muscles, that's endergonic. It takes ATP to make that happen. ATP hydrolysis is coupled with the process of muscle contraction. Muscle contraction will not happen unless ATP hydrolysis happens first. It's not spontaneous muscle contraction. There has to be free energy provided to make it happen. ATP hydrolysis provides the free energy needed to make muscles contract. They're coupled together. One won't happen without the other. Make sense? Coupling, not complicated either. All right, the main thing in this chapter, though, after we get through some of that energy stuff, is enzymes. And I'll tell you, coming up next week, we'll be back in the lab most of the week, sort of like we were with the fusion osmosis stuff. Give you a preview of what's going to happen. You've got one lab you'll do with enzymes that you'll have directions that say do A, B, C, D, E, F, G, and you'll just see what happens. You will need your lab notebook next week. Because from here on out, everything's going to go in that lab notebook. And I'll tell you how I want it done closer to that point in time. But you need your little lab notebook. First lab, you'll have directions, do this, do this, do this, do this. The second part of the lab, you're going to have to design your own lab around enzymes. And for that lab, you'll design the lab, you'll run the lab, you'll collect the data, and you'll do a poster presentation, sort of like we did with that first lab on that one. I'll probably do that next week. So be ready. The first lab will kind of give you some general guidelines about how you might want to do it, and then you'll go from there. But let's talk a little bit about enzymes with the time we have left. When you hear the word enzyme, I want you to first think about what they're made out of, which is almost always what? Proteins. Which means they're coded for by DNA. Which means if you have a mutation, what might happen? You might be missing an enzyme, or you might have a deformed version of an enzyme. Which means you're probably not able to do what? A particular what? A particular chemical reaction. That can be really bad. That can kill you. Tay-Sachs disease, for example, is a disorder, an enzyme disorder, that occurs in what group of people mostly? Tay-Sachs disease. Not Asian. Jewish. And it's a disorder where they can't metabolize what correctly? Certain fats. And because they can't break those fats down, what ends up happening to those fats? Their body stores them in a large part in the brain. And they end up going blind by like age 1.5 or 2. And they die by 3 or 4. There's no treatment, there's no cure. It's a death sentence. Well, there's lots of genetic disorders that tend to run in certain races of people, right? Like in the black community, what's the one? Sickle cell anemia. In white European people, what's the one? It's almost exclusive to white European people. What's the genetic disorder? It's called cystic fibrosis. Almost occurs completely in white European people. Tay-Sachs and Jewish people. Cystic fibrosis is the one where you make mucus that's really thick and viscous, and it ends up, you basically drown yourself.
What is MS?
MS is multiple sclerosis. And they're not really sure what's going on with that. It's a problem with your nerves. The coatings around your nerves harden and they don't conduct electricity right. But in terms of what causes it, not 100% sure. It's one of those things you just don't know about. But back to what I said, why do we have these racial genetic diseases? These genetic diseases originated from what? Where did they come from in the first place? Somebody mutated. And guess who the original mutant for sickle cell anemia was? A black person in Africa. And typically, historically, black people have reproduced with other black people. So what has happened to that gene? It stayed in that group. Okay? The first person to have sickle cell or cystic fibrosis was some white European person. And that gene has stayed in the white population. Tay-Sachs was probably originally a Jewish person that had that mutation, and it stayed in that group. That's sort of inbreeding, right?
Not the bad type.
Well, yeah, it's sort of, right? It turns out not to be good for genetics. But that's why we have racially based genetic diseases, and that's why incest is a really bad idea, right? Because we all have our own little freaky mutations. And most of us, other than Brittany, turned out pretty normal. Okay?
That was uncommon.
But why do we turn out to be normal even though we have mutations?
Because
Normally we're diploid. So even if we have a bad version of a gene, the good version usually covers it up and we're okay. But if close relatives are having babies together, you probably have the same freaky mutations, right? And your child might very well end up with two versions of those freaky mutations. That's why things like blue people started showing up in Kentucky, right? You ever heard of that? Google it. Google it. We'll have a lesson when we get to genetics. Real thing. Real thing. This has occurred in Kentucky. It's occurred in places in India where people.
I knew that it was racist when you said that about Brittany. I knew it.
I didn't even think about that.
We were like, wait, don't
Yeah.
Brittany is married their cousin.
She's not blue. She's not blue.
Whatever.
But I'm talking about people with very blue skin. The first time, yeah, if you've ever been to Kentucky, certain parts of Kentucky make South Alabama look like metropolitan. And that's saying something, right? There are some really redneck backwoods places in Kentucky. West Virginia is the same way. But in the backwoods of Kentucky, there are places not too many years ago where there weren't any doctors. And there was this group of people in a small town in Kentucky that literally had blue skin. The first doctors that saw them saw what? What would you think if Amy showed up and she had a really blue face? What would you think about Amy? Why might Amy turn blue?
She wasn't breathing.
She's not breathing. And you would think she's dying. So the first doctors saw these blue people and they came in and they thought, "Man, you're sick. What's wrong?" And they started doing all kinds of tests on them. Turns out they had a genetic mutation. And it was a recessive mutation that only showed up because this was a little backwoods town in Kentucky. And there weren't very many people there. Which meant, when you went out to find a wife or a husband, who'd you marry? Someone who was related to you. Maybe they weren't your sister or your first cousin, but they might have been your fourth cousin or your fifth cousin, or somewhere down the road. And eventually, this mutation showed up because people got two copies of this recessive gene. Now, as far as I know, in that town in Kentucky right now, there aren't any more blue people. I got online. I researched this years ago. One of the grandsons of one of the blue people is still alive, and he's heterozygous for the trait. He's not blue, but he has a gene for this trait. So if he was to marry another relative of a blue person, they could have blue babies. Now, if you know anything about India, And I'm going to defer to Brittany. What's the blue god in India? I don't know the name. Is it Krishna? There is one of the most powerful gods in Indian mythology. Is blue. Okay? Maybe largely based on the idea of this genetic condition that's not that rare in India. Yeah.
I don't think a lot of cult technology when people still believe in it.
Yeah.
Okay. Okay. I'm sorry. Okay. Indian religion. Blue people. I mean, think about it. 2,000 years ago, if you were here, and all of a sudden, this blue person shows up. What might you think? You might think they're an alien. You might think what? They're a god. You don't have a way to say. I mean, when the first Europeans got over to the Americas, what do people think? They're gods. I mean, that's why the Aztecs kind of gave up, right? How many white people did it take to conquer the Aztecs? I mean, it's like a joke, right? It didn't take very many people, right? Because what they do? They gave up because they thought the gods were against them. I mean, if you thought God was against you, you probably wouldn't fight too hard, right? That's kind of what they thought. There's no way the Aztecs could have, I mean, the Europeans could have beaten them otherwise, right? There was a lot of Aztecs. Yeah.
Wait, I don't know if this is true. It's probably just a myth. Do black people really have an extra bone in their foot?
Not that I know of. I don't think so.
Like somebody said it a couple years back that that's the reason black people passed.
No. No. I mean, it's a lot like this idea that myth that a lot of people think women have an extra what? An extra rib. Is that true? No. Okay. We have the same number of ribs. And that all comes from the idea that God created Eve by taking a rib out of Adam. But let's say that I wanted to create something from Kari, and I cut him open and pulled out a rib. Are his children, his male children, going to have less ribs? No. Okay? There's no basis to that. No basis at all. We have the same number of ribs. Hey, what you got? Thank you, ma'am. Janelle and Kari. No problem. All right, back to what I was saying. Let's talk a little bit about enzymes today, and we'll get deep into enzymes next time. Another thing that I want you to think about when you hear the word enzyme is their catalyst. If I said that Tessa over there is a catalyst for evil, what does that mean? So she just causes evil. Where she not only makes it happen, she also makes it happen how? Faster. Okay? So she can help to start it, or she can help to speed it up. Okay? That's what catalysts do. That's what enzymes do. They make reactions happen by decreasing the amount of free energy needed to make that reaction happen. They decrease the activation energy or the free energy needed to make a reaction happen. We'll talk about how they do that later. But one thing you need to know about enzymes is they lower the amount of energy needed to make a reaction get started. They lower the amount of free energy needed. Now, let's go back to proteins for a minute. We know enzymes are proteins. So therefore, their shape is essential. Structure determines function. And we know that shape is maintained in certain levels. Let's review the 4 levels of protein structure for a minute. Most basic level is primary, coded for by your DNA. And primary is essentially what order, what are in amino acids. And we know that in the primary structure, these amino acids are held together by what kind of bonds? Peptide bonds, which is a type of covalent bond. And it's a bond between what functional group on this one and what functional group on this one? Amino on one to carboxyl on the next. That's primary structure. Secondary structure It's how are certain parts of this chain shaped. What's the name for a chain of amino acids? Polypeptide. How are certain segments of it shaped? Might be in a helix, might be in a sheet. But once it gets in that shape, what holds that shape in place? Hydrogen bonds between polar R groups of amino acids. Tertiary structure comes next, and that's how each chain rather, is shaped. Before we get into the bonds that hold it there, why does it bend up in the shape that it bends up in? Hydrophobic amino acids want to be where? Away from water. Hydrophilic polar amino acids want to be toward water. So it bends so that can take place. In a cell, where does that bending and twisting happen?
In the rough ER?
Where does it happen? It can happen in the rough ER. If it's a cell that doesn't go through the rough ER, if it's a protein that is made by one of those ribosomes that's free floating, where does it bend and twist? What are those little tubes we talked about that help to bend and twist the proteins?
Histones.
That is stones. Called chaperonins. Protein tubes that help to bend and twist protein in the right shape. But regardless, tertiary structure is held into place by what two main kinds of bonds? Disulfide and hydrogen. And in the last level, quaternary, is how do multiple polypeptides twist around each other to form the overall shape of the protein. That's held together by all kinds of forces. Regardless, when we talk about a protein, there's 2 or 3 words we need to make sure we know. I want to get to a picture. Let's talk about this picture here. This is supposed to be a protein. Each one of those little spheres in there would represent what? Amino acids. It's a big group of amino acids all bent and twisted into some shape. There's a particular part of this enzyme that has a very specific structure. Here we're calling that part the active site. That's the site where what's called the substrate is going to attach to that enzyme. What is the substrate? What's the substrate? It's the substance an enzyme acts on, works on. For example, what's the enzyme in your saliva that we talked about? Amylase. That's the enzyme. What's the substrate of amylase? What's the thing it works on? Starch. It takes starch and breaks it down into simple sugars. So the substrate is the substance an enzyme works on, acts on, breaks down, or just as likely what? Build up. Enzymes are not all destructive. Some enzymes are catabolic, but just as many are anabolic. So here's the deal. This is an important thing to know about substrates and active sites. Their shapes have to what? They have to match. And not only their shapes have to match, what else has to match? Shape and charge. Shape and charge. You need to know, you need to write it down somewhere, that the shape and charge of the active site have to be complementary to the shape and charge of the substrate. Yep. The shape and charge of the active site have to be complementary to the shape and charge of the substrate. They have to match. A lot of times we describe this match between those two as a lock and key type of fit. Lock and key. The substrate and the active site fit together like a lock and a key. Now let's think about something. If they fit together like a lock and a key, how many substrates does an enzyme typically have? Only one, right? Specific. Each enzyme only works to do one reaction. What does that mean about the number of different enzymes you have in your body? got a bunch of them because each one is only good for one kind of thing. There is no jack of all trades enzymes that can catalyze multiple reactions. They do one. Usually just one.
So in case one quits working, it's not something
Well, not so much. I mean, you have multiple copies of each one. You don't have just one amylase molecule. You have millions of them. It's just so that, for example, your digestive enzymes, they're coded for by your DNA, right? What do we know about the DNA in your hand and the DNA in your stomach? It's exactly the same, which means the genes for making stomach acid or stomach enzymes are in your fingers. Would that be a good idea? Probably wouldn't be a good idea to have stomach enzymes in your finger, right? But the DNA's there. Why don't we have stomach enzymes in our fingers? Genes are turned on and off. The genes that make stomach acid, or stomach enzymes rather, are off in your fingers, but on in your stomach. There are other genes on in your fingers that are not on in your stomach. If we ever figure out how to do that outside the body, that's when we'll figure out how to make like organs for people, like replacement parts. We're going there. Like make if Christina needed a kidney one day, the goal of some scientists is to be able to make her a kidney. Not a kidney she has to worry about rejecting, but a kidney made with what? Her own DNA, so that her body wouldn't know the difference.
That's no.
Right.
Outline.
I think they will. I don't think it'll take that long. One of the ways they're trying to do that is by using what kind of cells? Stem cells, which are cells in your body that haven't really done what yet? They haven't decided what they want to be when they grow up. Okay? Stem cells. Which means they have the ability to what? Be anything. They could become stomach. They could become a brain. They could become whatever. What do you mean? They have used stem cells for certain things, like leukemia. Leukemia is a problem with what? It's a kind of cancer, but it's a cancer of what part of your body? It's a problem. Leuco means what? White blood cells. Leukemia, you have too many white blood cells and not enough red ones. So it's a problem with what part of your body? No? Where does the problem originate? Where's your blood originate? Where's your blood come from, Bill? Your bone marrow. Okay? The bone marrow. Sorry, I didn't hear you. Okay. Anyway, so what they figured out they can do, like for example, let's say that one of you guys had a child many years from now that has leukemia. Some people in that situation will then have another child to help the first one. How did they do that? Yeah. What do they take from the second child to help the first one? Not the bone marrow usually.
What's a little house?
They take the blood from where?
Their bones.
From the umbilical cord. The blood from the umbilical cord has a lot of what in it? Stem cells. And they take that stem cell blood and transfuse it into the child with leukemia, and some of those stem cells become what? Functional, working, what? Bone marrow. And they fix the sick baby. All right, it take a while. Something they started doing, and if you have a child, you can pay somebody to do this for you. There are banks that do this. They'll take umbilical cord blood from newborns and they'll store it and keep it, so that if that child ever needs it, or if a sibling of that child ever needs it, it's there in a refrigerator somewhere, and they can go get it. Well, probably not, because the name tag, the markers, are different, right? And Austin's child would probably reject that.
Oh,
His immune system would attack it.
if we do that, we're just hoping that our child has.
Well, you're just being cautious. You're preparing for the future, just in case. They might eventually use it for research, but I think it's thrown away. Unless you tell them they can use it. No. I mean, it would be lost in the placenta anyway. It would just come out of the mother and thrown away. I mean, most of the time what happens to the umbilical cord in a normal birth is it's thrown away. All right, now something else I want you to see here, and this is going to be an important topic for next class. For a long time, scientists thought the shape of this active site was hard, like the shape in a lock. What they found now is that it's not as set as they thought it was. For example, let's say you went today to Walmart and you got them to make you a key, like a house key. And you got home and you stick it in the lock and it doesn't work. How many of you have done that before? I've done it a bunch of times. Go got a key made, get home, go got it. That was terrible English. Stick it in a lock, and it'll fit in a lock because the shape's similar, but it won't turn. Well, most people would just throw it away, and because getting the key made for $2 or something, $2 or $3, and then just go get another one later. I'm not that way, I want to make it work. So if you want to make that key work, what do you do? If you want to make that key work, what do?
You jam it.
No, you take it and you hold it up beside the key that does work. You compare. You hold them side by side. You're looking at what? You're looking at the little grooves on it. Find where the grooves are a little bit off. What do? You take like a file. Grab it. But you slightly change the shape of the key so that it'll fit. Yeah. Smart. Regardless, here's what scientists have found happens. The substrate fits into this active site. Their shapes have to be really close for that to work. Their charges have to be really close for that to work. But once it fits, notice these arrows are trying to show you that the enzyme does what? It like slightly changes shape. What do you think that enzyme's probably doing to that substrate there? It's like giving it a hug. What do you think it's trying to do? Probably that wasn't just one substrate. That was probably two different things that fit in there. And when those two different things fit in, when that enzyme does this, what's it doing to those two things? It's pushing them together so they'll what? So they'll react, they'll bind. Other enzymes, once that substrate is in there, they'll kind of go like that. What do you think those are doing when they bend out like that? They grab onto the substrate and then they're trying to put torque on it in order to break some bonds, right? Catabolic kinds of things. That idea is called an induced fit. Now, normally when we talk about the shape of a protein changing, we call that what? Denaturation. And that's a big change in a protein, right? That's a bad change. This is a small change in the shape of a protein. A small change that makes the enzyme what? Work. This is a good thing for the enzyme. It's when the substrate binds to the active site and the enzyme slightly changes shape so that it will function. It may push two substrates together so they'll react. It may pull on one substrate so that it will break and come apart. Induced fit. It's a slight change in the shape of an enzyme so that it will function better. Induced fit in the shape of an enzyme so that it'll work better. I'm going to back up a couple slides and end today by talking about this next slide right here. This picture. I bet if you went home and you asked your parents, what enzymes do, what are probably 9 out of 10 of them going to say? No, I think most of them would have some idea of what an enzyme is. Most of them are going to think they're involved in digestion. Right? They break down your food. It's probably the answer most of you parents will give you. They break down your food. Is that true? That's true. But there are just, I want you to get through your head, there's just as many enzymes that help to build things up as there are that break things down. What are some enzymes you might have in your house that your parents might use and don't know they're using them? Probably not toilet bowl cleaner. Probably not toothpaste. Let's say you have a stain on the knees of your blue jeans. Maybe you fell on the grass. You got grass stain on your blue jeans. What do your parents put on there? Some kind of stain remover, like Shout, or I mean, there's lots of different versions of that. OxyClean's a little different, but a lot of those stain removers have enzymes in them to break down stuff like grass stain. Cooking at home. What enzymes might you have to help you cook at home? Is vinegar just an acid? Meat tenderizer. Parents ever use meat tenderizer? Good point.
Like an old man that was nasty looking here. Probably was a pedophile. I was crying, came over. Okay, the thing was like right here, so it's awkward enough.
Right.
Well, put some meat tenderizer on that. Sorry. Thank you.
But he probably actually kind of knew what he was doing. Because from the jellyfish, what's making you sting? Some kind of protein. Guess what meat tenderizer is? It's an enzyme that breaks down proteins. Okay? That's why when you put it on steak, it starts to digest the steak before you eat it, which means the steak gets wet. And it's easier to chew. So the whole jellyfish doesn't make sense. The meat tenderizer would deactivate some of those proteins, and this thing goes away. Makes sense. Anyway, up here. We're going to sort of make an equation out of what you see up here on the screen. I want everybody to focus, first of all. Up here, a lot of enzymes are involved in degradation. We haven't used that word very much. What words have we used for this kind of reaction? Hydrolysis, or when we're talking about metabolism, we call these catabolic. Okay, catabolic, hydrolysis, degradation. Most of the time, they're going to be exergonic reactions. They're breaking bonds. Notice what happens. The substrate will fit into the grooves made for it. The shapes are complementary. The charges are complementary. They fit together. So we've got an enzyme plus substrate. They eventually fit together. When they're together, we call that the enzyme-substrate complex. That's where the magic works. That's where the enzyme does whatever it's supposed to do. It catalyzes some kind of reaction. But then they separate. We get an enzyme plus a product. Maybe more than one product. But what you ought to see, that enzyme's the same everywhere, right? Which means it's not what? It's not changed. It's not used up. It helps to start a reaction, but doesn't actually take part in it. So that means what about your enzymes? They can be used over and over and over again. They're not actually used up. They're recyclable. That's a hydrolysis type of reaction. There are other enzymes that do more dehydration or condensation or synthesis types of things. And we see that here. The substrates fit into the grooves. The grooves line those two substrates up so that they will what? So that they will react. We still get an enzyme and a product. So this equation still holds. Enzyme plus substrate. We get an enzyme-substrate complex. And then we get an enzyme plus a product. Let me go back to my useful little Cracker Barrel toys here for a minute. Let's say that here is an enzyme. It's like the active site. It's got a specific shape. And I am making a substrate. Notice those will fit together, like so. Maybe. Let's say these are floating around in a big tub of water. One enzyme, one substrate. We're trying to make them react. If there's one of these and they're floating around in a big tub of water, what are the odds they're ever going to find each other and fit the way they should? Pretty small. What can we do to increase those odds? What could you do to that tub of water to increase the odds that the substrate and the enzyme will get together? You can heat it up. Heat means what? These little things start moving faster. Before chemicals can react, they first have to what? Find each other and what? Important point. Before two chemicals can react, they first have to collide with each other. But they don't just collide, they have to collide when they're turned the right way, right? If they collide like that, is anything going to happen? Probably not. They have to collide like that so that they're lined up. So one thing you can do to speed up reactions is to heat it up. Things are moving more quickly, therefore they collide more often, and the odds that they'll collide the right way are increased. It would make more sense, though, if we wanted this reaction to happen, maybe instead of heating it up, just wet. If we had more of this, if we increase the concentration of substrates, there's going to be a lot more chances that they'll find the right enzyme, right? So we can increase the concentration of substrates. We can also increase what? The concentration of enzymes. Either one of those is going to speed up the reaction, right? We can heat it up. We can increase concentrations of either the substrate or the enzyme. Can we heat it up infinitely? Eventually, we're going to heat the solution up enough that this protein is going to do that, right? And we call that what? Denaturation. So we're going to get to a point where heat's going to be a bad thing. But at first, unless we're already at our optimum temperature, heating it will help. And then there's a certain point where there's too much heat, and that's a bad thing. All right, I think that's a good place to stop.
Yes,